Yeah.
All right, Jeffrey.
Thank you. I'm presenting today my joint work with Shiv Kale and my advisor T.M. Murley at Virginia Tech on network-based gene function prediction for pathogenic bacteria. And normally people end with their funding sources, but to set the stage for my research, I want to start with mine. I'm funded through the FungiCat, or Functional Genomic and Computational Assessment of Threats project. This is organized by IARPA, which is the Intelligence Advanced Research Projects Activity government organization. And the main goal of this project is to identify the functions of genetic microbial sequences and classify their potential as threats. This has three main sub-goals, which are, for a given sequence, we want to identify the tax of origin, the functions or Go terms that sequence could perform, and then the level of threat, either to humans or agriculture, things important to the government. And our team is a large collaborative effort, which is centralized at Virginia Tech and includes some more well-known names, with Ido Friedberg at Iowa State, Daisuke Kihara at Purdue, Gaurav Pandey at Mount Sinai. You may have heard a flash talk from his group earlier today, and researchers from San Diego State University. And our group's focus is on Go term predictions. And the goal of this project has inspired us to take a fresh look at the question of Go term prediction in microbial sequences. So what I'm going to describe is our work with 19 pathogenic bacteria. We selected these based on them being highly clinically relevant, phylogenetically diverse, and have varying levels of functional annotation. And our main question is, how well can we make Go term predictions for these bacteria? And we're considering three main groups of annotations, primarily experimental, which are annotations within experimental evidence codes, such as inferred by a direct assay, computational evidence codes, which have been reviewed by a curator, but are based on computational analysis, such as inferred by sequence similarity, and then electronic, which haven't been reviewed. And to answer the question of how we can make predictions, we're using two main evaluations. One is to use experimental annotations to predict computational annotations, and then to use experimental and computational to predict electronic annotations. And I won't have time to get into the second one, but if you look at the annotations across all of these 19 species, you'll see they fall into sort of three main groups. At the top we have species that are well studied experimentally. We have those that have some experimental annotations, but primarily have computational based annotations, and those with little to no curated annotations. And the approach we have to make function prediction goes back to the early days of function prediction, which is using functional linkage networks. These are graphs where each node corresponds to a gene, and each edge connects two genes and may share a similar function. And these networks can be constructed from diverse sources of data, and there are many papers which showed how to build high-quality functional linkage networks to drive function prediction. Today I'm starting with a simple approach of using a sequence similarity network, where for the 19 bacteria we are using, we performed an all versus all blast. And in the case of reciprocal edge weights, we retained the weaker of the two, and converted the e-value to a score using the negative log. And we're using multiple e-value cutoffs to evaluate what kind of effect they have on our predictions. And the number of edges ranges from 700,000 to 2.5 million for the cutoff of 0.1. And then to make predictions for using our multi-species network, we adopt a term-by-term approach, which is where for every Go term, we add context by either labeling the nodes as a positive, a negative, or an unknown. And for negatives, we're using proteins that have annotations to other Go terms that are neither a descendant or an ancestor of the current Go term we're making predictions for. It's a simple approach we're starting with. And we're comparing multiple algorithms to the baseline of blast average, which is, in this case, simply the weighted average of direct neighbors in the network. Gene Mania, a well-known method, uses propagation across the entire network. And sync source is a method developed by Murley's group back in 2011. It's similar to Gene Mania, but it doesn't allow the scores of positive and negative nodes to change. We're also comparing to a newer method called Berg rank. This stands for Birelational Graph PageRank. And this makes predictions simultaneously for each protein by incorporating the Go hierarchy into the propagation. So we wanted to compare how that does. And Berg rank and blast average do not use the negative examples, whereas Gene Mania and sync source do. So the question we have to compare these are, would propagation of experimental annotations beyond immediate neighbors be beneficial? And we're using, as I mentioned, experimental annotations to predict computational annotations. The reason for this is that computational annotations of a protein must come from experimental annotations of some other proteins. And to give you an overview of our evaluation scheme, we start with the multi-species network and the annotations to a particular Go term. And then we run propagation with the experimental annotations, and then evaluate how well we're able to recover the computational annotations. And we repeat this for every Go term. And I'll show you some of the results now. So here I'm showing a box plots of the Fmax values. Fmax is the harmonic mean of the precision and recall, the maximum harmonic means. And then for every row, I'm showing the distribution of Fmax values for the 334 Go terms that had anywhere from 50 to 1,000 experimental annotations across the 19 species. And you'll see here, two of the network propagation methods, sync source and Gene Mania, have a higher median than blast average, and there is a significant improvement. We also see a wide range of Fmax values, and we wanted to dig in a little deeper to see how often are the network-based methods improving over blast average, and by how much. So here I'm showing the histograms of sync source and blast average, where the frequency is the number of Go terms, frequency on the y-axis is the number of Go terms in a particular Fmax bin. And at the bottom, I'm showing the difference between sync source and blast average. And you'll notice that there's about 10% that have an equal score between the two methods, 10% where blast average does a little better, but the majority, we see an improvement for sync source over blast average, with 10% of the Go terms having a difference greater than 0.2. And we see similar trends for Gene Mania. I'm just showing the results for sync source now. We also wanted to ask, these results are from using a e-value cutoff of 10 to the negative 25, which is relatively stringent. And we wanted to see if we still see an improvement using network propagation with less stringent cutoffs. So focus on the top two histograms as I step through the different cutoffs. You'll notice that they both shift to the right, with blast average seeming to catch up a little bit. So stepping back now, I want you to focus on the bottom histogram. You'll see that the difference does come closer to zero, but we still see, for the large majority of Go terms, we still have an improvement using the network propagation. So looking back at the box plots, we see that almost the median for these different methods all have Fmax median less than 0.8. And we hypothesized that the computational annotations that we're missing may be coming from experimental annotations of species outside of our original 19. And as almost a kind of sanity check, we developed a method to mimic the inclusion of experimental annotations of other species by including the computational annotations in our predictions. And we used a leave one species out validation method. And what that means, we start with the annotations of a Go term once again in our network, and then leave out all of the annotations of one of the species and use the computational and experimental to run network propagation. And then evaluate how well we recovered the computational annotations in the left out species. So we repeated this for every species with Go terms with at least 10 computational annotations. And here we see a significant improvement of the Fmax across all the methods. These are 561 Go terms with at least 10 computational annotations. And all of them have a median Fmax of above 0.9. There's less of an improvement with network propagation, but it's still there. And we still see a really wide range of Fmax values. So we wanted to dig in a little bit and understand what's going on and for which Go terms do we do more poorly. So we dug into each of the individual species, and you'll see that for two of the species, H. influenza and S. typhimurium, we have a really high median Fmax above 0.95 with good performance for almost all Go terms. And for S. typhimurium, we see that there is a statistically significant improvement of network propagation over blast average, which is why I have it bolded. So now looking at all of the 9 species that had enough computational annotations, 6 of them had a statistically significant improvement for network propagation. And if you look at the top 7, the median Fmaxes are above 0.9 for all these species. But for Vibrio cholera and Mycobacterium tuberculosis, we have a lower median Fmax and a wider range down to 0.8. So we wanted to look at what's going on with those species. And if we look at how many computational annotations these species have, you'll see that Vibrio cholera has a lot more computational annotations than the other species, which may make it harder to make predictions for it. So that can explain why we do a little worse for that species. But for Mycobacterium tuberculosis, we don't see that difference. So there, one of my first thoughts was maybe it's more phylogenetically distant than the other species are to each other. And if we look at a phylogenetic tree, we see that Mycobacterium tuberculosis is the only one in the Actinobacteria phylum, which can confirm that it is more distantly related to the other species than they are to each other. So in summary, I used multi-species sequence similarity network to predict computational annotations from experimental annotations. And we saw that network propagation performed superior to blast average, irrespective of the e-value cutoffs. And we also saw that including computational annotations from other species mimics the inclusion of experimental from other species and improved our validation results. And we have seen other papers in the past that have looked at the effects of using different cutoffs in co-expression networks and saw that when using a less stringent cutoff, just the local neighbor method can do about as well as network propagation. But here we still see an improvement for network propagation. And some of the next steps are we're planning to compare to other algorithms, including top performers in CAFA. We're working to integrate multiple types of data, including string networks for as many species as we can. We're working to improve negative examples, as we heard from some talks earlier today of how to improve the negative examples there. And we're also making progress on scaling up to make predictions for all proteins in UniProt. And I've also invested a considerable amount of research in speeding up our methods in preparation to make predictions for this massive network. I was able to run the entire leave one species out pipeline in under 15 minutes. So I have a good improvement there, and hopefully it'll work for the huge network. And finally, I'd like to thank Murli and Dr. Kale, and as well as my funding and the NSF Travel Fellowship that helped me get here. And thank you. I'll be at the poster spot 185. So thank you. Any questions?
Any questions? I have questions. So, given the incredibly high, in CAFA, BLAST never has a Fmax above 0.3 something. And the best performing methods didn't break 0.7. So given that in your testing, BLAST has a really high Fmax, and the other methods are only incrementally better. If that, the question is, how hard is the problem you're actually presenting if BLAST is performing so well? How difficult is the function prediction if BLAST is doing such a great job? And BLAST is supposed to be like the lower limit on things. And so that's my question.
Yeah, so we are using, when we're computing the Fmax, we're just using the negative examples rather than all proteins that don't have the specific Go term, so that does improve the Fmax of BLAST average and also for the other methods. Also compared to CAFA, we aren't evaluating the experimental annotations. That's a more difficult problem than recovering the computational annotations, which are generally sequence.
CAFA is not evaluating the experimental annotations either. CAFA is using them as a baseline. CAFA is,
right. Yeah.
So CAFA is definitely not evaluating. There is no need to evaluate experimental annotations. They are the gold truth.
Oh,
experimental annotations are the gold truth. So you don't evaluate experimental annotation. You use them, and you evaluate, whatever other types of annotations, right? Actually, you don't evaluate annotations. You evaluate predictions.
Yeah. Yes.
Can you talk about the algorithmic or computational innovations that led to you being able to run these in 15 minutes? Sounds impressive.
Yeah, sure. We had, looking at the propagation method, it uses multiple iterations to compute the scores of each of the nodes across the entire network. And we saw that to get a accurate score for each of the nodes, you had to use oftentimes many hundreds of iterations. And we developed ways to perform these iterations faster by using a sparse matrices in our computations, which helped. And we also compared if we use less iterations, how close is our score to the final score that we would get if we had used all the iterations completely. And we found that in general we were able to get close to like 95% of the scores of the ranks of the proteins correctly if we only use a small number of iterations, like 20. So that led to us being able to run these much faster.
Okay, so move to our last speaker. So, we'll see